Hi, I am Dr. Yanzhe from California Institute of Technology, presenting our paper published in ACS Applied Biomaterials entitled Single-Cell Phenotypic Analysis and Digital Molecular Detection, Linkable by a Hydrogel Bead-Based Platform, authored by myself, Dr. Jing, Dr. Xingyu, Dr. Xiao Huang, and Professor Michael Hoffmann. The motivation for developing this whole platform was the widely observed phenomena of cell heterogeneity. It basically describes that clonal cells from one culture can behave differently. Because the cells are dynamically changing all the time, to better probe the underlying molecular mechanisms, we want to develop a high-throughput analysis tool combining hydrogel and droplet microfluidics to potentially conduct in-situ molecular analysis following single-cell viability phenotyping. Here is the schematic of this work. We first developed a very simple disposable droplet generation device, then established assays for single-cell viability phenotyping, digital PCR, and digital LAMP. We finally developed the phase transfer and reagent exchange recipe for linking them. For hydrogel chemistry, we chose to work with polyethylene glycol cross-linking between four-arm acrylate and dithiol. These monomers are highly biocompatible and the cross-linking is spontaneous. This recipe has been used in a bulk membrane form for cell culture, for MDA and LAMP amplification reactions. The cross-linking reaction is fast and spontaneous and does not need any cross-linking initiation that may harm the cells or nucleic acid to be analyzed. To be able to make hydrogel beads with encapsulated cells or DNAs, we need a droplet generation method that is fast to finish droplet generation before the gelation even starts, and hopefully also disposable to avoid biochemical contamination. We designed a centrifugal droplet generation device using a needle with a bent tip, put in a microcentrifuge tube. The aqueous phase is added to the luer lock of the needle, and the oil phase is added to the bottom of the tube. With centrifugal acceleration, the aqueous phase can be forced into the oil phase and get pinched off into droplets floating at the top of the heavier fluorinated oil. We experimentally found the droplet size and its coefficient of variance were tunable by changing the needle inner diameter, the oil volume at the bottom, and the centrifugal acceleration. We also developed a simulation model to study the effect of other physical parameters, like the bending angle of the needle has limited impact on the size of the generated droplets, and lowering the interfacial tension by changing the surfactant itself or its concentration can further decrease the droplet size. Please see the supplementary movie for our droplet generation simulations. And then to establish assays inside the beads. First, in the figures on the left, by counting the fluorescent cells encapsulated inside the gel beads, we verified that the cell distribution pattern followed Poisson statistics and was not much affected by the low centrifugal acceleration used. For the viability phenotyping assay, a cell proliferation dye, Alamar blue, was added to the aqueous phase. The dye can be reduced by actively metabolizing cells into a form with bright red fluorescence, so that the bead fluorescence can act as an indicator of the insider cell viability. The quantitative performance of gel bead viability phenotyping was verified by analysis of observed fractions of bright fluorescent gel beads, as shown in the figures on the right. Effective single-cell phenotyping in gel beads was achievable within 4 hours for Salmonella type B. For molecular analysis assays within gel beads showed enhanced compartmental stability during the heated process, including PCR and LAMP. We then established efficient assay recipes. Starting with PCR, these amplification and detection processes involve lots of ions, small single-stranded or double-stranded DNAs, dye molecules, and proteins. In the figures on the left, we found that the gel bead digital PCR was slightly inhibited, likely due to the reduced diffusivity in the cross-linked hydrogel network. By supplying the DNA polymerase, the largest component that was likely affected by diffusivity, the amplification performance was boosted to be comparable to that of droplet digital PCR standard. And figures on the right showed that with modified recipe and serial diluted target DNA loaded, the Gelbeads delivered digital PCR performance as good as droplet digital PCR. For digital lamp and Gelbeads, as shown in the figure on the upper left, the negative beads containing no target DNA were able to remain individual beads with no false positive, but the positive beads with targets were mostly aggregated together, leading to severe cross-contamination. This was likely due to the large amount of amplification products. Those negatively charged amplified fragments may change the interfacial tension, then cross-linking may attach the beads to the neighboring beads. This problem was solved by adding BSA proteins, which is a protein commonly used as a surface coating to prevent adsorption. Using the modified GD lamp recipe, Gelbeads were able to lay flat as one layer for imaging, and the neighboring positive beads did not have the radial pattern originating from the interface, shown in the lower left figure. With this recipe, we further established amplification efficiency curve with DD lamp, using serial diluted DNA load, and demonstrated a good match. Finally, in order to demonstrate our platform's immediate potential on linking phenotypic and molecular analysis, we also developed the reagent exchange protocol for in situ PCR following viability phenotyping. The reagent exchange process involves first breaking the emulsion with a chemical demulsifier to collect the beads carried in the aqueous phase, and then drain the liquid and perform a freeze-thaw treatment to enlarge the surface pores. And then incubate with concentrated PCR reagents for infusion, and finally drain and resuspend in oil phase for in situ PCR. With this protocol, we qualitatively demonstrated successful in situ PCR, showing that the PCR reagents were successfully infused into Gelbeads for target amplification reactions to proceed inside. Overall, in this work, we have developed a single-cell analysis platform using PEG hydrogel beads, generated by the disposable needle-in-a-tube device. For cell viability phenotyping, we showed the highest single-cell encapsulation rate under Poisson distribution and viability phenotyping assays using cell proliferation indicator dye. For digital PCR and digital lamp, PEG beads survive the heating better, and the concern in amplification efficiency can be solved by slightly modifying the assay recipes. The reagent exchange between the phenotypic and molecular analysis was demonstrated feasible. With a bead sorting step added after phenotyping, hopefully this platform will generate new knowledge with a linked phenotypic and in situ molecular analysis. With that, we would like to thank the Bill and Melinda Gates Foundation for the funding support, and thank you for listening and your interest in this work.